Hi, I'm Deborah Zaritsky. I'm reporting for Dr. Valentine, Chem 530, Grand Canyon University. Today is December the 2nd, 2020. Today I'm reporting on the receptors tyrosine kinases and how they influence cancer. This is a two-parter video, so please make sure that you stay tuned for the next part. Our tyrosine kinases are going to be responsible for many cellular activities, metabolic activities in particular, processing insulin, and we're also going to look out at those tumor-causing genes as well. So those receptor tyrosine kinases are also known as RTKs. So if you do see that abbreviation, that's what that's going for. Humans do have around 60 of those receptors that do aid in that signal transduction. So we are going to be looking at the how the communication is going between that intracellular fluid and that extracellular space. So our tyrosine kinases produce signals outside of the cell by ligand binding domains and spread those messages from plasma membranes and enzyme active sites on the cytoplasmic side. So also our TK has signaling pathways that I'm going to be mentioning here that prevent uncontrolled proliferation and contribute to cell death or apoptosis. There's multiple types of kinase groups that I am going to mention and that do contribute to the occurrences of various cancers. And I'm also going to be talking about some inhibitors that contribute to the treatment of cancer. So there are very many kinase members. So we do have approximately 58 of them. As I had mentioned, there's 60, so we just kind of round up though. Scientists have not been able to pinpoint exactly how many there are. We do know that they transfer the phosphate group utilizing energy from the ATP there. So those tyrosine kinases are classified as receptor tyrosine. They could also be the IR, which are the insulin receptors, and they could be non-receptor tyrosine kinases. So we do have about 20 subfamilies, and I've only listed a few here, but they're pretty significant. Before I touch on those, I would just like to further explain that the ligand binding region in the extracellular domain is a single transmembrane helix and the cytoplasmic region. So I'm going to be referring to a picture on the next slide, so just kind of keep that in mind. I'm also going to be mentioning the EGFR, which is identified as class 1 in the slide, and the ERBB, which is the the epidermal growth factor receptors. So that IR could also be the FGFR. I'm also going to be mentioning class 5, which is a VEGF, or vascular endothelial growth factor. So when we're talking about these signal pathways, we are going to refer to them as cascades because they are multi-steps. One kind of leads to another, and we're going to see how this can be found in the blood vessels and also the epithelial tissue within the stomach. So according to Maruyama, the total number of tyrosine residues found in each intercellular region and the number known to be modified differ significantly between superfamily members, and the distribution of tyrosine residues is quite variable. When comparing the insulin receptors, as I had mentioned, the IRs, to the RTKs, Myaruma says the RTKs are all expressed as single protomers that form non-covalently associated dimers. Dimers I'm going to be referring to in just a moment. The IR, or the insulin receptors family, consisting of IR, the insulin-like growth factor 1 receptor, or IGF-IR, and the insulin receptor-related IRR, is also expressed as a single subunit, but it undergoes processing into form 2, which is alpha and beta. So this polypeptide chains that are assembled into a heterotetramer, or alpha B squared, homodimer stabilized by disulfide bonds. So what that looks like is this. So what we have is our signal molecule binding site. So I'm referring to the picture on the left. And then we have our alpha helix within our membrane. So that signal molecule binding site was in our extracellular space. So we have our alpha helix in the membrane. And then we have our tyrosine kinase regions of protein. So those single molecule binding sites are made of monomers. And then once we have our chain of events, then those monomers are gummed together to become a dimer. So all of those families that I had mentioned are really important because all the RTKs have a single transmembrane domain that separates that intercellular tyrosine kinase region from the extracellular portion. The RTKs can be triggered by hormones like insulin to stimulate the transcription of particular genes. So on the right, we would have a signal molecule that would then activate through the alpha helices and the, forgive my pronunciation, the juxtamembrane domain. So when this occurs, we have the phosphorylation of the tyrosine. So signal transduction is started by a sequence of growth factor, or the ligand binding, to the extracellular domain, as I had mentioned, and then that dimerization of the receptor proteins and the autophosphorylation of the receptor. So the receptor autophosphorylation creates phosphorylated tyrosine residues on the cytoplasmic molecules. And it is the combination of the signaling molecules that determines the specificity of individual receptor protein tyrosine kinases. So Nelson and Cox kind of said it well, that in addition to the many receptors that act as protein, the tyrosine kinases, or the RTKs, the several receptor-like plasma membrane proteins have protein tyrosine phosphatase activity. So the picture here depicts that juxtamembrane I was having trouble pronouncing. So the kind of anatomy of our ligand binding site here is we would have the extracellular domain where we are having our signal molecules attach, and then we're moving on down through that domain to the kinase domain, where ultimately we're going to get phosphorylation through those various amino acid combinations. And that tyrosine kinase domain does include an activation loop whose orientation and phosphorylation determines the active or inactive site of the kinase domain. So that intracellular juxtamembrane region participates in an autoinhibitory interaction that must be disrupted for tyrosine kinase activation. So to target those receptor tyrokinases, signaling pathways remains a challenge for scientists and clinicians working in the cancer field. Several small molecule inhibitors and antibodies are being clinically developed to target these RTKs. So it's called the MAP kinase and the P13K-8KT pathways, according to Riggin. So the way it's going to work is those signal molecules that I was referring to, they could be a carcinogen that's coming from, any cancer-causing agent that we come across. It hits our normal cell, and it's going to get these genes called the proto-oncogenes or oncogenes that are going to change the conformation of our proteins. So once these are activated, then it's creating a clone, and then those cells are just going to proliferate, inhibiting the apoptosis. So around 30% of the RTKs are mutated or overexpressed in various human cancers. The oncogenic mutations or gene duplications in the juxtamembrane that I mentioned, in that region of the KIT and the FLT3 result in the constitutive activation of these receptors in the absence of their ligand and are consequently directly linked to the carcinogenesis process. And that's according to Davis, Wu, and Nurkiewicz et al. There was a protein that I had mentioned on the first slide, the second slide, and it was the ERBB protein. So it belongs to a family that can control the development of the neurodegenerative disease or the defects in embryonic development of the lungs and the skin, the heart, or the brain. And then the VEGF family that I had mentioned, they allow permeability in the blood vessels. So when these are overexpressed or mutated, they're also going to express a certain kind of a growth. So mutations in the RTKs or proteins that facilitate their cascade or their pathway, signal pathway, have been implicated in the onset and progression of a wide variety of diseases. We talked about those IRs, and that can create the insulin resistance and diabetes. We have inflammation, bone disorders, atherosclerosis, angiogenesis, and then I'm going to talk about some the gastrointestinal stroma tumors. So these genes are can cause many abnormalities. An example of one of these abnormalities is also ovarian tumors and lung carcinomas, and those are caused by the mutated EGFR5. So these strong associations that I had mentioned with the EGFRs and its primary ligand, the EGF and TGF alpha, are expressed in many human cancers. And as I mentioned, that's the lung carcinoma, bladder cancer, breast cancer, and the glioblastoma. So I'm just giving a little bit of statistics here according to Paul and Mukopade. Okay. So as I had mentioned, it's just a visual here of the various kinds of cancers that can be expressed by these various receptors. So by definition, cancer is when we have a single cell and it's dividing without that control and that regulation by the RTKs. And they produce many clones of those, and so it just grows and grows and grows into other spaces. So cancer is generally the result of an accumulation of mutations in oncogenes and tumor suppressed genes. The oncogenic defect can have many proteins involved. I did enjoy when we were working on our discussion questions, and it was very interesting to see how the mutation of just one receptor can cause several similar symptoms or signs as another mutated receptor. So with that being said, the protein kinases, there's several different kinds, but one in particular called the CDK is in charge of the cell cycle. So the progression through the cell cycle is regulated by the cyclin-dependent protein, CDK, which act at specific points in the cycle for phosphorylating key proteins and modulating activities. So that going back to that EERB2 oncogene that I had mentioned in previous slides, which is the ERBB protein. It's a shorter version of the EGF, or the epidermal growth factor. This protein lacks the extracellular binding site for the EGF. So in one of the classes that I had mentioned in the first slide, one was the VEGF. Yes. Sorry, I had to, did I say too many E's in there? So the VEGF plays an important role in the proliferation, migration and vascularization of the endothelial cells as the most powerful vascular penetrant. So my little visual here is it dependent upon the amino acid getting phosphorylated or the protein, it has a different dimension or a different avenue that it's going to be taking, ultimately leading to the angiogenesis. So just giving you a moment to look at that picture there. As I had mentioned, that when we do have cancerous cells, they're just going through their mitotic activity without any regulation, right? There's no rules here. So they just grow and grow and grow, impeding upon the healthy tissue. And that's what the gastrointestinal stroma tumor stromal tumor is going to look like. So it's characterized by the activating mutation in genes coding for that RTK. So this is where the RTKs are definitely going to influence cancerous activity. So these mutations are predictive, there's one in particular that I wanted to mention, and that was the, and pardon my pronunciation again, the imatinib mesylate. We're finding that this is acting as an inhibitor, which is going to go ahead and block that ligand site, and then we will not activate that receptor. So here it is again, that imatinib was, I thought endoplasmic reticulum was bad. The imatinib was developed as a tyrosine kinase receptor inhibitor, and it was previously known as a family of compounds called the trifositins, that were shown to have a specificity to that epidermal growth factor receptor that I had mentioned, that EGFR. And the tyrositins are just a fancy word for the small molecule of the TRs. So we also have another kind of inhibitor, and that's going to be a monoclonal antibody, and that's called the MABS, and that's going to block the ligand binding site. And so that's the picture that I'm showing you here. So if we have our ligand represented by that little green oval or ellipse, is showing that we don't want that ligand binding to the site there. So we would have that monoclonal antibody as an inhibitor, and that will not activate the RTKs. Hence, we would have normal survival or cell migration. So the monoclonal antibodies block the binding of the ligand, avoiding conformational changes needed to activate the tyrosine consequently, and the activation of the cascade pathways. So those cascade pathways are going to be blocked, and we will not continue to proliferate those abnormal cells. And here are my references. As you look at Zhao on the next page, I did find an interesting quote from him in regards to the VEGFR expression. And it said that there's a significant positive correlation between the VEGFR expression level and the degree of the vascularization and malignancy of tumor tissue. So it has been my pleasure doing research for this presentation. And thank you for watching.